Yes, student. Again, this is very important sequence of the content. What is it? Purification of colloidal solution. Student, this is not only a content but it is a sequence. If you watch all the video in sequence, you can better understand it. That is what before this video we have checked how to prepare lyophobic sole. So we have seen there to prepare lyophobic sole, we require to add a small amount of electrolyte in freshly prepared precipitate and then the colloid will form. Okay, that means what? To formation of a colloid, strong electrolyte required but its quantity must be limited. Getting? This electrolyte is responsible for the formation of colloid. But the main thing is what? Its quantity must be what? Limited. If its quantity is in excess, then besides the formation of colloid, the colloid itself will getting precipitate out. Okay, that is what? Coagulate. That is, there will be the destruction of colloid. Okay, so what to do? If we remove this excess quantity of strong electrolyte, which is, I use the term here what many times, crystalloids. It is used as crystalloid. So if we remove the crystalloid, that is excess quantity, so that only limited quantity of crystalloid will present with colloid. This is important for its stability. This process is known as for purification of colloid. Now, what to do? One of the method is there, dialysis. In the dialysis method, in a vessel containing what? Large amount of water and a dialysis bag. A dialysis bag is this type of structure made up of animal bladder, animal membrane or parchment paper. Parchment paper. It consists what? Large number of very small pores in it, but it is too much small in size. Now inside there is a mixture of crystalloid, crystalloid. Crystalloid means what? Electrolyte. Okay, and colloid. And what? Colloid. Now the colloid particles are big in size, while the crystalloid particles are small in size. These pores are also having very small size. So only crystalloid molecule can move from the parchment paper to the water kept outside. Thus, the limited quantity of crystalloid is removed from the mixture of these two. Okay, this is called what? The process of diffusion, and this process is known as dialysis. We can improve the technology, that is, if we have to do the process very fast, we can apply here what? Electric field. If we apply electric field, this is called what? Electric dialyzer. In electric dialyzer, the positive field and the negative field are applied. Now, if the crystallite consists ions, suppose it consists ions, then the cations and anions are present, like Na cation, Cl anion, or Fe cation, Cl anion, etc. ions are present, then they can move very fastly to opposite electric field. That means the process can be easily done in short period of time. One more purification process is known as centrifugation. Centrifugation means what? Rotating mixture of colloids and crystalloids. Again, crystalloid means electrolyte at very high speed. So, if at very high speed it is getting rotated, now the colloids are big particle. I told you their size is 1 nanometer to 1000 nanometer. That is, they are heavier as compared to the electrolyte, that is crystallite. So, being they are heavier, when it is rotating at very high speed, the colloids particle will settle down due to gravity, while the lighter crystallite will remains in the surface, over the surface. Now the whole mixture is decanted so that the lighter colloids crystallite particle will move out. Okay, this is called ultra centrifugation process.